Good mid-afternoon, everyone, and welcome to today's Mid-Afternoon Masterclass, one of the marquee events of the University of Melbourne's 10th Annual Science Festival as part of National Science Week. My name is Julia, and I will be your host for today's masterclass and all the masterclasses happening every day this week at 2 p.m. As the host, I'd like to acknowledge the traditional owners of the unceded lands on which we are filming, the Wurundjeri Woi-wurrung people. Aboriginal and Torres Strait Islander people are this country's first scientists with deep and enduring knowledge of the land, waters and skies. I pay my respects to elders past and present, and I welcome all First Nations people here today. In making this acknowledgement, we commit to respectful and responsible conduct towards all others according to the traditional laws of this land, particularly at times of formal ceremony. So today's event is How Sneezing Chickens Impact the Egg Industry with Dr. Sathya Arachchige. Sathya came from Sri Lanka to study at the Melbourne Veterinary School. During her PhD, she explored bacterial respiratory infections in chickens using advanced molecular tools. Her postdoctoral research investigates upper respiratory tract infections in Australian free-range chickens using next-generation sequencing. And out of the lab, Sathya loves hiking and is also very much into wildlife photography. So welcome, Sathya. Thank you so much for being here today. I'm very excited to hear all your presentation. So how today is going to work, Sathya will give her 20-minute-ish presentation. Then we will be taking questions from the audience, and then we'll aim to finish about 2:45. And also, heads up to everyone, we are obviously in a lab today, and so there are a few noises, a few noisy machines around, so apologies if there is a little bit of background noise during the event. To ask a question, we will be going to questions at the end, and so to ask a question, just put the questions in the Q&A function. We have that open. There'll also be a couple of polls during the event, so watch out for them. Also, we've got the chat open, so any comments or maybe clapping or cheering emojis, whatever you'd like to do. We welcome all that, but please, of course, be respectful in the chat. Now, to make sure everything is working as it should, could everyone out there in the audience let us know, open up the chat, let us know where you are zooming in from, whether it's a suburb, or a city, or a country. We know this is an international event and we'd love to find out where you're all zooming in from. And it's already coming in. Okay, we've got Indonesia, hello, welcome, Melbourne, Fiji, Point Cook, Sri Lanka, so many places, a lot around Melbourne, Carlton, amazing. Thanks everyone. Welcome to everyone, Iran as well. Oh, I love this, so international. Okay, awesome. Great, well thank you so much. Without further ado, Sathya, I'll hand over to you. Thank you.
Yeah, thank you. Thank you for that kind introduction, Julia. And thank you everyone for joining with us for today's masterclass. All right, so let's get started to see how sneezing chickens impact the egg industry. And before going into details, I would like to ask two questions. Okay, the first one would be, have you noticed different types of eggs in supermarkets? So I would like to see your answers.
So the poll is up on the Zoom for everyone to answer. I'm going to answer as well. So the answers will come up. Okay, so we've got a 98% for yes. That is a lot of people have noticed it. So
so that's really great.
Yeah.
And then I'll show you some. So I have brought 3 types of eggs that I could find in the supermarket. So this one is called cage eggs, and the cage-free eggs, and also the free-range eggs. So out of these 3 types, so if you have noticed these 3 different types of eggs in the supermarket, which one do you usually go for? So can you answer that question?
All right, so the poll should be up online as well. So we've got free-range, cage-free, caged, and no preference.
Yeah.
I'm going to answer myself. There we go. Okay, so the answers should be coming up pretty soon. Make sure you put all your answers into the poll. Let's see. What do you predict? Oh, okay. All right, here we are. So, okay, which type do you go for? Okay, so we've got 57, go for free range, 15 cage-free, 15% caged, and 23% no preference. There you go. Is that what you expected?
Exactly. So
Okay.
that really matches with the statistics from the last financial year.
Okay.
So it sounds like more than 50% of the sales, egg sales in the supermarkets are for free-range eggs. So that's what we saw here as well. And I'll explain a little bit about these three types of eggs. So if you see cage-free, so that means so these chickens are coming from cage-free chickens. That's why they are called cage-free eggs. So these chickens are kept in large sheds, so they can freely roam within the shed, but they don't have access to outside, right? And the caged eggs are coming from the chickens who are kept in cages. And the free range, so that's the one that I'm going to talk about in detail today, because we work on free range chickens in Australia. So within last 15 years, I think the demand for free range eggs has been increased, so that the free range operations in Australia has gone up as well. So I'll talk a bit about who are free range chickens. As I said previously, as I mentioned briefly previously, so these chickens have regular access to an outdoor range during the daylight, so they can go out whenever they want during the daytime, so they can freely roam around, interact with each other and forage, and also they can come inside if they want. And these chickens are housed securely and comfortably in sheds at night. And so as humans, these chickens also get lots of diseases. So here is one example, which was in news all around a few weeks ago. So bird flu, which was found in many couple of free-range farms in Australia, in couple of states. And with this incidents are emerging up, so we saw that there are people were scared there'll be an egg shortage in the supermarkets, and some supermarkets, I think they had to put a cap on the number of egg cartons that you can buy from the supermarket. So that's how these diseases can impact in many different ways. And out of all these different types of diseases, I'm going to talk about respiratory diseases specifically, because our project mainly looking at respiratory diseases in free-range chickens in Australia. And when it comes to respiratory disease, it's kind of like a mixed salad. So there are bacteria, there are viruses, there are fungi which cause respiratory diseases in chickens. And the interesting thing about these respiratory diseases is that most of these diseases are caused by multiple organisms at the same time. So that's why it's like a mixed salad. So I'll show you some pictures to show how these sick birds look like when they have respiratory diseases. So as you can see in these images, they get eye discharges, nasal discharges, and swollen eyes and sinuses. Sometimes they keep their eyes closed all the time, and they become so lethargic, and they just lie around. So that's not good for them, actually. And so what is the impact of these diseases? So as we talked about the bird flu incident, so when these chickens are not well, so they are not happy, and they do not produce eggs as the healthy chickens do. Not only that, the number of eggs go down. At the same time, the quality of the eggs can go down as well. So when the number and the quality of the eggs go down, so there will be losses to the farmers. And also, not only that, so when the chickens are not well, so they are not happy and they are not comfortable, so that will decrease the welfare of these birds as well. So when it comes to these impacts, so what are the solutions that we have? So and how can we minimize these impacts? The very important thing is early detection of these organisms that cause these respiratory diseases. And also when it comes to respiratory diseases, as I said, previously, so these respiratory diseases are caused by multiple organisms at the same time. So it's very important to detect all of these causes to come up with good control measures and also good prevention methods to prevent future infections. So that's not easy, and I'll take COVID-19 as an example to show how we diagnose different diseases. And I'm going to ask another question again. So if you can remember, I'm sure you might have done lots of COVID-19 testing within the last couple of years. So if you can pop in, what types of tests have you undergone for COVID-19?
So do you remember how COVID-19 was detected? That's the poll. So we got the rat, PCR, and genome sequencing.
Yeah.
Oh, okay, it's all gone. That was quick. Okay, let's see. All right. So we've got 59% said the rat, 39% said PCR, and 2% genome sequencing.
Yeah, so that's what we expect, because I think the most popular test is the rat test. And some of you might have gone through PCR testing as well, but not genome sequencing mostly.
Yeah.
And I'll explain. So COVID-19 was caused by a virus. And we can detect this virus by growing it and identifying it. And also we have these serological tests like RAT. So what happens is we take a blood sample from the patient and look for proteins, either the proteins produced by the patient against the virus, or we can detect the parts of the proteins of the virus in the blood as well. And in PCR, what happens is we look for a specific region of the genetic material of the virus. So if the PCR detects that part in your sample, so then your PCR returned a positive result. And also, time to time, if you can remember, there were different variants of the virus emerging. So how we detect these different variants of the virus is that we take the whole genetic material of the virus coming from, let's say, 2 different patients or 2 different countries, and then we sequence the whole genome, and then we compare the genome sequences. And if they are identical, so we identify them as the same variant. If they are different, so then they become 2 different variants of the same virus. So then you can understand, so even for just one virus, we had, multiple diagnostic tests. So imagine how tricky when it comes to respiratory diseases in chickens, because there are multiple organisms causing respiratory diseases in chickens at the same time. So if we did that many tests for one virus, so then how many tests do we need for all these pathogens? And also, if you can remember how much time and how much efforts took to come up with those diagnostic tests against the COVID-19 virus, so it's not easy. So when it comes to the respiratory diseases in chickens, if there are bacteria, so we can grow the bacteria and see what's in there, and we can do virus growth, and also we can do different serological tests for each pathogen, and also we can do specific PCRs for each pathogen. And not only that, we can do genome sequencing to identify these bugs as well. But so doing multiple tests for multiple bugs is not that easy, and it's time consuming, it's labor intensive, and might be expensive as well, because we have to do lots of testing. So in this project, what we are trying to do is we are trying to use metagenomics. It's a molecular tool to see, instead of doing multiple diagnostic tests, to see if we can catch all the fish, if we could fish all of them in one go. So that's our aim. And I'll explain in a bit how we do that. So what we do is we take a piece of tissue sample from the respiratory tract of chickens, and So when we take that kind of a tissue sample, it contains the chicken cells, so the ones in blue, and also the microbes. And then from all these cells, we extract the genetic material. So that contains both the genetic material coming from the chickens as well as coming from the microbes. And we sequence them all together. And then we remove the sequence reads coming from chicken genetic material as much as we can. And then the remaining sequence reads we map to a collection of microbial genome sequences available in different databases to see what do we have in our sample. So that's basically what we do, or what we are trying to do. And so when it comes to research, it's not always, very smooth and you get what you expect. There are lots of challenges on the way, and you have to experiment different things to choose the best, the process or the pipeline that works for you. Here is one example. As I mentioned previously, when we take a tissue sample from chickens, so we have to deal with both bugs, the genetic material coming from the microbes, as well as the genetic material coming from the chickens. So there are different sequencing technologies. So one is called nanopore sequencing technology, and there's another one called Illumina sequencing technology. So I can show you this small device. It's a tiny, very handy small device. That's the nanopore sequencing device. So we have this facility in our lab and all the other equipments and things that we need to run the machine. So that was there's no much infrastructure needed to run this machine, and it's fairly rapid. And as I said, so we have the facilities to do that in our lab. And when it comes to Illumina, so it's a fairly big machine. You need more infrastructure, and it takes a longer time to process, and we don't have Illumina sequencing facility here. Of course, our first choice was nanopore sequencing, but not Illumina. But so this chicken genetic material interfered with nanopore sequencing technology, so we had to go with Illumina. So that's just one example, the challenges that we have to face while doing this kind of research. And this is a snapshot to show what we have achieved so far. So in this table, the first column shows the organisms that we were able to detect by metagenomics. And the last three columns show what are which organisms were detected by other different diagnostic tests. So you can see that metagenomics detected all the organisms that were detected by other diagnostic methods, and some more as well. So that were not, we were not looking for those organisms because either they were not culturable, or we didn't have a specific diagnostic test for those organisms. And so just a summary, where have we come so far? So as I said, so the metagenomics detected all the organisms detected by other diagnostic tests, and detected some other organisms that are difficult to grow, and detected organisms for which specific diagnostic tests are not available as well. And interestingly, we detected some organisms that are unknown to cause respiratory infections in chickens, because we were not looking for those organisms because they are unknown to cause respiratory diseases in chickens. So what's next? We are fine-tuning this method to differentiate, because when it comes to commercial poultry farms, there are vaccines that we use against certain pathogens. So when we do sequencing, because if they are live vaccines, that means we use a live organisms, but they are not pathogenic as the pathogenic strain. So they live in the chicken, stimulating the immune system. So when they come across those pathogens, they provide protection against that particular pathogen. So when we get these sequences out of the chicken tissue samples, there could be vaccine, the sequences coming from the vaccine strains as well. So at the moment, it's a bit difficult for us to differentiate between the pathogenic ones and the vaccine strains. So we have to fine-tune our pipeline, and we have to come up with more deep analyzers to differentiate the vaccine and pathogenic strains. And also, since we have detected some pathogens which are unknown to cause respiratory diseases, we really don't know what they do. Are they just sitting there, or do they play a role in respiratory disease in chickens as well? So that's an area where we need to explore more. And also, at the moment, so sequencing is not that cheap. It's a bit expensive, but it's becoming cheaper over the time, because it's been using for lots of research at the moment. So it's becoming cheaper with time. So, but we have to come up with a more cost-effective and less time-consuming way to use it as a routine diagnostic test. Like if you want to use that in the lab and provide results to the field, we need it to be very quick. And so all this research work, it's not individual work. So there's always a very big team behind. And I would like to give a big shout out to Amir, Mark, and Chaturi. So they are the key people who are working on this project. And also, when it comes to research, you need a bag of money as well. So the funding for this project is coming from Australian Eggs. And so the Scholexia is helping us with providing the samples from the field. And in addition, the Stephen Wilcox from VHI. So that's where I do my Illumina sequencing, because we don't have facilities here. And in addition, I would like to thank the university and the Asia Pacific Center for Animal Health. So our lab is part of that big research group in the Melbourne Veterinary School. Yeah, so that's it. And I'm happy to take any questions.
Amazing.
Yeah.
Well, thank you so much for that presentation. It was such a good overview of all the work you do, and yeah, such a good background. So thank you. And I mean, thank you for all your work that you're doing as well. It's so important. And probably something that we don't realise is so important to be done as well. So yeah. Thank you. Yeah, so we do have some questions. So please put the questions in the Q&A function in Zoom. So we'll keep trying to get through as many as possible. We've got about 20 minutes to go. So let's start going with some questions already. Now, our first question, which I actually really love this question because it actually involves yesterday's masterclass as well. So those of you who tuned in yesterday and today will really be interested in this one. So the first question is, can chickens have tuberculosis?
No.
No, there you go. Okay.
Not chickens.
Okay. Well, there you go. That was definitely something that we were talking about yesterday with tuberculosis. So thank you for that question, CGK. Okay, so we've got a few questions about bird flu and humans. So is bird flu harmful for humans? From Divya.
There are some, again, when it comes to bird flu, so that's caused by the influenza virus. So there are different types of virus, and some of them are pathogenic to humans as well. Yeah.
Very good. Okay, so off to Joshua, Joshua MK. So how many farms have been affected by bird flu, and can they die from it?
When I heard about it last, it was 7 farms.
Okay.
Maybe the numbers has gone up.
Okay.
Yeah.
Okay, there you go. And Joshua, follow-up question. How do scientists know if an egg is infected?
Oh, okay. So it's it's a good question.
Yeah.
So it's difficult to say, by looking at the egg, it's difficult to say it's infected or not. But there will be some changes in the egg shell and the color and things. But again, it's not just infections. Even nutritional deficiencies can cause changes in the appearance of the egg.
Yeah.
So just by looking at the egg, it's difficult to say whether the egg is infected or not. But so what we do is we can take a sample and do further testing, and so that's how we usually do. That's the confirmatory way if you want to know whether the egg is infected or not.
Yeah. Okay, very good. You mentioned the nutritional value as well, and so I wonder whether the different types of eggs you mentioned right at the start, are there any different nutritional values that come through? Do you know?
Not really, because these chickens, no matter how they are raised, so they are fed on a similar diet.
Yeah, okay.
So, but when it comes to free-range eggs, so they have the opportunity to roam around and forage. So they might pick some insects and things, which other types of chicken will not have the opportunity to. But that doesn't make much difference. Yeah.
Okay, there you go. So it's more about the welfare of the chickens rather than the actual nutritional value.
Exactly.
Very good.
Yeah.
Okay, so on to Anonymous, that's fine, you don't have to tell us your name. So yeah, I think we're talking a little bit more about, I guess the process of the experimentation. So what organs are tissue samples collected from, and why are those in particular?
Okay,
Yep.
so this project, so this project is specifically looking at the upper respiratory tract infections in chickens. So if you take the respiratory system of chicken, so you divide that mainly into two, like upper respiratory system and the lower respiratory tract. The main organs in the upper respiratory tract are the nasal cavity, the trachea, the sinuses, and the conjunctiva. And we take samples from all those four sites.
Okay.
And at the moment, we are looking at the things in the nasal cavity. So that's where we see lots of changes. So, yeah.
Yeah, okay, very good. Well, good question, thank you. So a question from Hayden now. So is testing for these kinds of illnesses compulsory or down to the individual farmer? And does your research rely on their input and I guess requesting their participation?
Yeah, it's not compulsory, but if you don't do that, so more chickens will get sick, and that will affect the production of the farm, and then that will affect the farmer as well. So it's not compulsory. So when they are sick, the farmers get really panic. So they want to know what's happening to my birds in the farm. So anyway, they will ask the veterinarian to come and have a look, and the vet will go to the farm and look at the birds, and then they will send the samples to labs.
Yeah, okay. So it's
to see what's good. Yeah.
Yeah, absolutely. Okay, good question. Thank you.
Yeah.
Okay, so one from Danielle. Are these diseases not an issue for caged and cage-free eggs? Would they therefore be recommended over free-ranged eggs?
Okay.
Yeah, so is there a difference, I guess?
Yes. So it's not like that. So these diseases can happen in any type of chickens. We are focusing more on in this project, we are focusing on free-range chickens because with the increase in demand for the free-range eggs, the number of free-range operations in Australia has gone up. So now it's mainly free-range, mostly free-range farms in Australia. So that's why it's becoming a major part of the egg industry in Australia.
Yeah.
So that's why we are looking at the free-range chickens at the moment.
Yeah, okay.
But this can happen in any type of chickens.
Yeah, okay. Yeah, gotcha. All right, good question. Thank you, Danielle.
Yeah.
Okay, so sort of biosecurity, and with the farmers as well, so interesting with all the farmer questions.
Yeah.
Dennis, what are the biosecurity measures farmers can take to reduce the risk of getting bird flu, and how do free-range farms manage this risk as well? Good question?
Yes.
Or do you know? Yeah.
So when it comes to free-range farms, that's actually the disadvantage, because these chickens freely roam, and so they can contract diseases from wild birds and things. So then it's very important to have strict biosecurity measures to prevent those infections coming into your farm. So you can have, like, you can control the traffic into the farm, like the people and the animals coming into the farm, and you have to be very careful about your feed source, the water, and you have to be, so those are the things that you can look at, and you have to be the strict biosecurity measures to prevent the contact between your chickens and the wild birds and humans as well, because they can bring the infections into the farm.
Yeah, that's true. So multiple ways that they can really be infected. Yeah, absolutely.
And feed and water as well.
Oh yeah, many things. Yeah, good question. Thank you. And also just a reminder, apologies for some of the background noise. As I said, we are in a lab, so there's some fridges going on and off in the background a little bit. Don't worry, it's not your computer that's making the odd noises. Thank you for the questions, by the way. They're still coming in. So really, really interesting. So one from Holly. Do infected eggs ever make it onto supermarket shelves, or is this way earlier in the process?
No.
Yes. Don't worry, everyone.
Don't worry about that. So there'll be inspections, and only the good quality eggs will come into the supermarket.
Very important to know.
Yeah.
We can safely go shopping tonight.
Yeah.
That's okay. Okay, a bit more with the vaccines now. So Nafis, thank you for the question. So how does the vaccine influence the composition of the respiratory microbiome in vaccinated chicks? Are there any observed shifts in commensal bacterial populations that might affect susceptibility to other respiratory pathogens?
Does the vaccine influence the composition of the respiratory microbiome? Okay, so...
Complex question.
Yeah. Okay, so these, if I talk about the live vaccines, it's actually the same organism, but it's attenuated. So it has been, the pathogenicity has been reduced. So it doesn't cause a disease in the chickens, but still stimulate the immune system so that it will protect the chickens against the pathogens when they contract the same pathogen next time.
Yeah.
So it might change the commensal bacteria a little bit, but there are lots of research going on to see if they change the microbiome or not. But I don't think there'll be, massive changes which can cause other things, because before we use a vaccine in chickens, there are lots of trials going on to see if it is safe and effective as well. So if any vaccine is causing something like that, then it's not allowed to use that vaccine.
Yeah, okay. Yeah, rightio. Okay, good question. Very good. Okay, so in terms of, I guess, the sick chickens and I guess infectious diseases, can the chicken sickness affect any other animals or people? That one's from Bryce.
Can
Thank you, Bryce.
sickness? So there are some pathogens like that. So bird flu or the influenza is one. So they can infect both chickens and humans and other animals as well. There are like organisms like Salmonella and few E. coli. So these pathogens, they can infect both chickens and humans as well.
Yeah, okay, so a few of them. Okay.
Yeah. So there are some organisms actually you can, if for example, if chicken meat or eggs were infected with that organism, and if people consume that, so they can pass on to humans as well. So those are called zoonotic diseases.
Yeah, okay.
But not all of them.
Yeah.
Only few,
Okay, all right. So some diseases, but then we've actually got a question from Kyra as well. So on this topic, so if birds evolved from dinosaurs, is it possible that bird flu could transfer to modern reptiles? Bit of an evolutionary question. Don't know much.
Don't know. Yeah.
Good question though.
Good
We should all find that out. Very good. Okay, I might change tack a little bit. We've got sort of 10 minutes, so keep the questions coming in, but I'd love to hear a little bit more about, I guess, your background and what led you into this field of work. So what's your background and why did you decide to study this field?
Yeah, so I am a veterinarian, graduated from Sri Lanka. So after completing my degree, so I wanted to, my interest was in research. So I was looking for opportunities, and then I came across this PhD opportunity at the Melbourne Veterinary School. And then I came here. So my only interest at that time was only microbiology. So I had no specific, interest in chickens or anything. If it's microbiology in any animal, I was happy. But then my PhD was about microbiology in chickens. So I was working on a respiratory pathogen called Mycoplasma gallisepticum, which is causing respiratory diseases in chickens. And that's where all this began. So after completing my PhD, I felt like, okay, I should stick to this and I should explore more. Yes, that's how it all began.
Yeah, fair enough. So what is it about this work that interests you the most?
So when the techniques, I would say the tools that I use and how fascinating and how the results come up after using those tools. So that, that's fascinating. Yeah,
very good. Okay, so we'll go back to a few more questions. Okay, so from Daryl, what does a sneezing chicken look like?
Oh,
okay.
So I showed you some pictures already.
Can you maybe go back?
Yeah. If I can.
Not very well, I think the answer is.
Yeah. They become so lethargic and they have lots of discharges coming from the eyes and nostrils.
back.
Yeah, back.
Quite early on.
Yeah, probably one more. Yes.
Okay.
So,
thank you.
And in chickens, actually, when there are respiratory infections, there's lots of happening in the eyes as well, because eyes and the nostrils or the respiratory system are like close to each other. So you see lots of changes in the eyes as well. That's why you see the eye discharges, the nasal discharges, and in chickens actually there are two sinuses just underneath the eyes. So they also get affected. So the sinuses become enlarged. So the face, then the face of the chickens look like a little bit fluffy, and and sometimes when their eyes are very severely affected, sometimes they can't even open their eyes. And they just keep their heads down and they just lie on the floor.
Yeah. Not good at all.
And there are some pathogens which can cause deaths of these birds as well.
Yeah.
So they just die.
Yeah.
Yeah.
So with your work, I mean, are you often in the farms, or are you really in the lab most of the time doing all the work with the samples?
At the beginning of the project, actually, we went into the field, and because now there are field veterinarians who are sending us the samples, but initially we had to go and train them to collect the samples the way we want.
Yeah, okay.
So initially we went to the field as well.
Yeah, okay. And so what does like a typical week of work for you look like now? Lab, office, meetings? What does a work as a scientist
It's a mix
look like
of
for you?
everything. Yes.
Yeah.
Some days it's just all meetings. Some days it's all in the lab, and sometimes when it comes to my work, so once I have the sequencing data, so it's lots of work on the computer to analyze these data.
Yeah.
So it's kind of like mix of everything, like office work, the lab work, meetings and things. And also I have a couple of PhD students who are working with me, so I have to help them as well. Yeah.
What's your favorite part of your work?
I think I prefer the time that I spend in the lab. Yeah. So that's the most important, like enjoyable time.
Absolutely. And so a question for, I know in our audience there's some younger sort of high school students possibly interested in coming into this sort of field. Do you have any advice for students of that age who are thinking about continuing to study science, go to university, study science? What advice would you give to those students that are thinking about going into that field?
I would say it's fascinating. There are lots of fascinating stuffs happening in the science. It doesn't matter which field, but there's lots of things happening in all the fields. So if you are interested in, or if you think of coming into science, I'm sure it will not disappoint you. Yes.
Amazing. Very good. Okay, well, I think we've done all the questions and we've had the presentation, so we might wrap it up there. Thank you so much for the presentation, for all the work that you're doing, and for sharing it all with us today. We really appreciate it.
My pleasure too. Thank you for giving me this opportunity to talk with everyone.
Yeah. Absolutely. Well, we really appreciate it. Okay, well, thank you everyone online as well for joining us. We appreciate you zooming in today for this week for National Science Week. So the mid-afternoon masterclasses will be held throughout the week. So if you'd like to attend another session, make sure you register via the link. There'll be something sent in the chat, but also there are more details on the Science Festival website. Tomorrow at 2 p.m, how to catch a giant coconut cracking rat. So there you go. And I know our first question combined our first two sessions this week, so maybe someone out there can think of a question that combined our first 3 sessions. That's the challenge for everyone. Okay, we'll say thank you so much. Thank you, Sathya.
Thank
and we'll
you,
see you
Julia.
tomorrow.
Yeah.
Bye.
Bye.